Okay, this is lecture 17, part 2. We're continuing looking at the adaptive immune response, which is specific immunity mediated by T cells and B cells, which have receptors for different antigens on each different individual cell has different foreign antigen receptor. On the B cells, it's antibody molecules, and on the T cells, they're T cell receptors. Now, the genetic process that generates these antigen receptors, either antibody or T cell receptors, is a random mechanism. It's a mechanism that randomly generates in each individual cell a different antigen specificity. And in a random process, you could get all kinds of receptors, including receptors to self. But the immune system normally is self-tolerant. The body doesn't make antibody against its own tissue, self. That would be an autoantibody, and it would cause a disease process. And the body also doesn't make T cells that have receptors that react to self. Again, it would be an autoimmune response. So we are tolerant to self. Otherwise, you would have essentially an autoimmune disease process, which is not that uncommon, but and is a problem. So in a normal individual, you are self-tolerant. Now, self-tolerance is generated by the process of clonal deletion. The clonal deletion involves destruction or killing of any self-reactive B cell or T cell clone that develops out of the immune system responsible for development, production, and maturation of B cells and T cells. So if you do get a B cell that has receptors on its surface, the antibody molecules on its surface that could react to self, they will self-destroy, they will be destroyed. And same goes for T cells. And the destruction is by a process called apoptosis. It is referred to as programmed cell death. Apoptosis and programmed cell death are the same process. Apoptosis is a type of cell death which does not involve any inflammation or inflammatory process. And so this is a normal regulatory process that destroys self-reactive cells of the immune system, of the adaptive immune system. And in that way, we are, if all things are working properly, then we are normally self-tolerant. As I said, the apoptosis, there are actually several different types of cell death that have been described, but these are two of the main ones. Necrosis has been described for many, many years, and that's when cells die. That's the situation when cells die of injury, and they get phagocytosed, and they'll also stimulate an inflammatory reaction followed by wound repair. Some sort of repair mechanism goes on. Now, in apoptosis or programmed cell death, the apoptotic cells get phagocytosed, they don't produce any inflammatory reaction. And this is the kind of thing that happens in processes that requires certain cells to die. Normal, regulated body processes. Okay, let's talk about clonal selection. And this is really the basis of how the adaptive immune response works. and how it adapts to the presence of a foreign antigen, a particular specific foreign antigen. And this process also applies to T cells, not just B cells, although there are some differences, of course. Well, in terms of B lymphocytes or B cells, the bone marrow produces the B cells and from stem cells, and the maturation process occurs within the bone marrow. And for this reason, the bone marrow is considered a primary lymphoid organ. It is the place where some of the immune tissue is produced and becomes mature and is exported from there. And each new clone of B cell clone is each new B cell is a unique clone which has a unique receptor on its surface and a unique antibody molecule on its surface. And it's unique in that the variable region, the antigen binding region, will have a single antigen that it can bind to. So each clone has antibodies all over its surface that have one antigen specificity. These are antigen receptors. The mature B cells leave the bone marrow and they migrate out into the blood and to lymphoid organs that they populate. If a specific antigen, if a particular antigen gets into your body, a particular virus or microorganism like a bacteria or fungi, or if some sort of molecule enters into you, it will activate the B cell clone in you that has the appropriate receptor for that antigen. It will activate the one or small number of B cell clones that can respond to that particular antigen because they have the appropriate receptor for that antigen. In other words, they have the appropriate antibody on their surface. And with activation, that B cell starts to proliferate. And two different types of cells will form with that multiplication of the cells. One type are called plasma cells, and they start to secrete that specific antibody. So you have dividing cells that are secreting the antibody with the right specificity. And the other type are memory cells, which are very long-lived cells that will respond if, and these cells settle down and don't divide and they just anymore, and they'll sit there, and they'll respond if you get exposed to the antigen much later. A second exposure, and another exposure to that same structure molecule that they're specific for. So you have plasma cells and you have memory cells being produced. What this means is that the appropriate clone of B cell is selected by the presence of the antigen to which it responds. This is the process of clonal selection. And this is why your immune system can respond quite specifically to particular foreign antigen, and the memory part of it, the memory cells, are why you become immune to that. So you can't, you will be protected the next time you get exposed, because if you are exposed the first time, you'll have memory cells. So the second response is much larger. There are now many, many more cells that can potentially respond. Response will be quicker, larger, and you will have no effects typically from the microorganism because you are immune. Clonal selection has pulled out from all the different cells in your body, and there really are billions of B cells and T cells. They've pulled out the right B cell with the right antigen receptor on its surface, the right antibody molecule that can respond to that, and multiply and produce B cells, and also memory cells that allow you to be immune from then on. And this is shown here in this diagram. Not, the numbers are not quite right because, I mean, obviously a whole bunch of antigen doesn't respond to this. There are millions, billions of B cells. They've drawn the antigen bindings antibody in a shape on the surface, but it's a drawing, it's an artist's interpretation of it. Truth be told, of course, there are many, many antibody molecules all over the surface of each B cell, and there are a few cells, small number, that can respond to a particular antigen, and those are the ones that start to multiply, and you get more and more of them. These are the memory cells, so you get plenty of memory, and you get a lot of B lymphocytes. You get a leukocytosis, not only of all the white cells, but particularly the lymphocytes. With enough time, you will have plenty of them as plasma cells that produce and secrete antibody into the plasma, and enters the tissues, goes into the lymph, goes throughout your body. Clonal selection. Now, antibody has many, many functions. The primary function, of course, is that I'm going to draw it in the abbreviated form, even though it's 4 polypeptide chains. I'll just draw it as a Y. The primary function, of course, is binding of a specific target antigen. But after that, then there are all kinds of other things that the antibody molecule can do. It'll bind, if you have it free floating around in you, it'll bind onto the target antigen, and then it can trigger all kinds of responses. Okay, so, yes, you have billions of cells in you, and some microorganism gets into you, and the innate immune system cannot stop the infection, so it's still there, it's multiplying. Eventually, some molecules from it will interact with a B cell, triggering that B cell to, if it has the appropriate antibody, that B cell to bind, start to multiply and proliferate, and secrete antibody, and you have antibody, being secreted in your body, and that really ups the game in terms of defense mechanisms. For example, antibody can bind onto the surface of bacteria and clump them together, and that enhances, which is called agglutination. So it can cause agglutination, and that'll enhance the phagocytosis activity of this. So phagocytic cells more easily phagocytose these kind of agglutinated bacteria. You can get opsonization, in which the presence of antibody on the surface increases the phagocytosis. It's not because it's clumped, it's because it's got antibody on its surface. Not like agglutination, it's different from agglutination. Opsonization. You have direct neutralization. It could be neutralization of a toxin, because the antibody blocks the toxic active site of the toxin. It could be because the antibody blocks the ability of the virus to bind to the surface of the cell and enter into the surface of cells that are its target. Or it could be it prevents binding, adhesion of bacteria onto cells that, bacteria also need to be able to adhere to tissues in order to cause an active infection and start proliferating. Some antibodies can fix complement. IgG can fix complement. IgM can fix complement. And with complement fixation, and this is via the classical pathway here, activation of complement. This is via the classical pathway. With that, you get C1, 4, 2, 3, and then 5B, 6, 7, 8, 9, forming the membrane attack complex that inserts into the membrane of the bacteria and allows contents to spill out. Another mechanism is that Um, with activation of complement, you get chemotaxis of cells moving towards the area of infection. And you also have another mechanism called ADCC, antibody-dependent cell-mediated cytotoxicity. I'm not going to go into describing that. There are other actual mechanisms. Actually, there's some other mechanisms that are important. Suffice it to say that agglutination, opsonization, neutralization, activation of complement, chemotactic inflammatory effects, and ADCC are all important ways in which are all important protective mechanisms that are activated by the binding of antigen to by its antibody, specific antibody. Okay, let's talk in terms of the antibody response. The very first exposure to an antigen shown here, this is time along this horizontal scale, and this is the amount of antibody. It's called a titer. So this is the ability to bind, really. So if it has a lot of binding activity, it has a high titer. And this is time. It's showing the development of the antibody response. When you're first initially exposed to antigen, it takes a few days before there is an antibody response. In this case, what we're looking at is we're looking at this red line here, and after a few days you start seeing some antibody, and the antibody is an IgM class specific for the antigen that the person is exposed to. You get an increase in IgM, and that's going to peak somewhere between 7 to 10 days. And then it starts to drop off, and as it peaked, just as it's peaking, the IgG response starts to appear. So the same specificity against the same target, but IgG. Now, the IgG response will form and start to be of some significance between 10 to 14 days, and then it starts to drop off. Now, in this case, IgM drops off and IgG will drop off. So the primary response, the very first response to an antigen is always IgM, and then followed by IgG. A secondary response occurs when you have another exposure, a second exposure. And in this case, they're showing the second exposure occurred here at, say, about day 24 or 25. Right there. But it could have been much, much, much later. It could have been even years later. But in this case, they're just showing that their exposure to the antigen again another time, once the IgG and IgM response drops off. Now don't forget, in that response, first IgM, then later IgG, what you got was not only an expansion of the cells that had the right antibody production, could secrete the right antibody, but also memory cells. And so with secondary exposure to that antigen, you get a rapid IgG response. The secondary response is going to be IgG, mostly IgG. And so here you see here at this point, and it was arbitrarily chosen. It could have been at a later time, but you have to put it somewhere, and so it can be observed. And here they're showing it at day, I think, 24. There's a rapid, very high increase, very high titer. And this is a logarithmic scale, this is the vertical scale. So very, very high production of IgG class antibodies that are specific for this antigen. So the primary response is IgM followed by IgG, and the secondary response is almost entirely IgG. There is some IgM response, but mostly IgG. And with time that will drop off. But again, you'll have memory cells, even more memory cells. It's the kind of thing that you get an infection and you fight it off. If you get exposed at some point, it doesn't have to be at this time point, at some point later, if you get exposed, you will respond quickly and you won't see any sign of an infection. That's the secondary response. The same idea occurs if the first initial exposure is a vaccine. With the vaccine, you get some component of the microorganism. You might even get inactivated, weakened sometimes organism, something that can go into you, can multiply a little bit but not much, just to give you exposure to the antigen. But you don't get the disease with the vaccine. But you do trigger your immune response, and you do get a primary immune response, and you do get memory cells, so that if you were exposed a second time somewhere down the road by infection, you would produce such a quick IgG response that you don't get the infection. You are immune due to vaccination. One of the most important discoveries and in development in health sciences that has ever occurred. It is the reason why millions and millions and millions of people are alive today, and their descendants. And some of you might not be here if your ancestors or you had not been vaccinated. It's a wonderful thing. You should note this, but you should also note that if you were given you were exposed at the very same time that you had the secondary exposure to this antigen, if you were exposed also to some other antigen, for that other antigen, it would be the primary. First time you'd seen it, you would get a primary type of response to that other antigen. You would not get a secondary response. You had only seen it one time. So you would get first the IgM and IgG. Okay? So that's a normal antibody response, primary and secondary. Primary antibody response and secondary antibody response. Now, one of the important developments that has occurred is the development also of monoclonal antibodies. These are produced from a single clone of hybridoma cells. What they do is they isolate a B cell that is secreting the antibody that they want, an antibody that has the right antigen specificity, whatever they're searching for. They're looking for an antibody to a particular antigen, and they find and they combine it with a myeloma cell, a partner. They hybridize the two together. They fuse them together into one cell. A myeloma is a B cell tumor that can grow and grow indefinitely. So in this hybrid, they actually physically fuse the two cells together. And in the two partners that make up that hybrid, the myeloma provides the immortality. So the cell line can grow and grow and grow in vitro. These are typically grown in vitro. On a commercial scale, they can be grown in giant tanks that are 2-3 stories high. The specificity of the antibody is provided by the normal B cell partner in the hybrid union. This partner provides the antigen specificity. So once they've isolated hybridoma product from a fusion of a myeloma cell and an antibody secreting B cell, they have an immortal hybridoma cell culture, and they can produce unlimited amounts of monoclonal antibody. And those monoclonal antibodies can be used as tools for research or investigation, or tests to see if the person has been infected or not. or sometimes treatment. There are many, many uses of such monoclonal antibodies. Very, very important discovery. Here's an example of showing how this is done. In this case, it's mouse. Now, mouse antibody is not the same as human antibody. Remember that antibody molecules are protein molecules, and a mouse antibody molecule is quite similar in its overall structure to human antibodies, but it still is different enough that it is not self. It is a foreign protein. If you injected that into a human, you would get, it's a foreign protein. You would get an antibody response. The human's immune system would, adaptive immune system would respond and produce antibodies against that antibody, that foreign antibody protein. Well, for that reason, if you want to develop a monoclonal antibody that can be used in a human, then there are ways that they can change mouse systems into human antibody producing systems. I'm not going to get into that right now. I might describe it later. Anyhow, just to give you an idea, if you're trying to produce a mouse monoclonal, you inject the antigen into a mouse, and after 10 or 14 days, you once you've gotten the full response, you might boost it again with a second exposure, so you get lots of IgG producing cells, and you isolate the spleen, which is full of B cells, and you make a suspension, and you put these cells together with myeloma cells and fuse them. Now there's a way to kill off any unfused myeloma cells and only have the hybrids, and those hybrid cells can then be placed one at a time into, it's very easy to do this at a huge scale. You can put them in one cell into each well on a multi-well plate. They sell these 96 well plates. You can have, if you have 100 of 96 well plates, you got 9,600. 9,600 different individual hybrid cells. You can get many individual ones and grow them up, and then test each supernatant for the presence of the antibody you want, whether the antibody, the desired monoclonal, is there. If you find it, here they're showing the desired one. Well, don't worry that it's coming out of an empty well. It's one of the wells that has a hybridoma in it. That's just a mistake, I guess, from the artist. Anyhow, you can identify the desired well, and in that way you can identify which cell had which cell culture is the one you want. I wouldn't worry too much about this. I'm not going to ask you a question on how to develop monoclonal antibodies. This is just for your own interest. Now, these monoclonal antibodies, as I said, can be used for multiple purposes. They can be conjugated to a toxin. Toxin can actually be bound onto the antibody so that it could kill targeted cells. If you have a monoclonal antibody against a certain cancer cell that's linked to a toxin, then the cancer cell, the antibodies will bind to the cancer cells and kill. The toxin will kill them. These are things that actually have not been that successful in human treatment for cancers, but let me get into something else which has been successful, and that is getting around the problem of the fact that the mouse antibody is a non-self, non-human antibody, and therefore the immune system of the human would react to it. What they have now are different ways of humanizing monoclonal antibodies so they can be injected into humans, and the human doesn't react to them by producing an antibody to them. human antibody against them. One way is to partially humanize them, that you take genes. Well, I'm going to go to I'm not going to describe these two. I'm going to describe the one that is the most effective and can fully humanize a monoclonal antibody. You can take mice that have a defective immune system themselves, and because they don't have a proper immune system, they will accept grafts, tissue grafts, from humans. And you can graft into these mice human bone marrow stem cells. So they'll produce human blood cells. Their blood system will be human. So when you inject an antigen into such a mouse, and they won't reject the human cells, the human B cells that they're producing, they won't reject them. So when you inject antigen into such a mouse, you'll trigger a human antibody response from human B cells. That's a fully humanized monoclonal antibody. And you go on to, choose to try to find the right one and produce a hybridoma that's producing the right antibody, monoclonal antibody. Single clone, antibody from a single clone. But it needs to be human. Otherwise, if it has any mouse protein characteristics, a human that you injected it into would mount an immune response against that antibody. Okay, let's talk about. We talked about clonal deletion and clonal selection for the immune response. There is also, and I don't want you to get confused, I realize the word selection is used again here differently, but there is a process called thymic selection that occurs, and during thymic selection, what you get are all the self-reactive T cell clones are being destroyed. So let me start at the top here. T cells, unlike B cells that get produced and mature in the bone marrow, T cells are initially produced in the bone marrow, but these are T cell precursors. They are not fully mature T cells. They migrate to the thymus. Remember, I told you that the thymus, I hope I told you, that the thymus, or the ones I taught anatomy and physiology too, the thymus is a primary lymphoid organ. Many textbooks and people who teach who don't understand immunology well enough, they talk about it as an endocrine gland. It is not. It is a primary lymphoid organ where the immature T cells produced in the bone marrow go to mature into fully mature functional T cells or T lymphocytes. So cells produced in the bone marrow, these precursors, go to the thymus, a bilobed gland sitting on the top of the heart, and the T cells mature there. Every T cell clone is unique from the next one. Each one has a T cell receptor on its surface, all over its surface, specific for a particular antigen. And they have a single antigen target specificity. So the T cell receptor is how the T cell interacts with an antigen, but only the one that it can bind to. Meanwhile, if any self-reactive clones are produced within the thymus, they are deleted. Okay, they are deleted before they leave the thymus and populate the rest of the body. Otherwise, any T cell that is matured and is not self-reactive, is tolerant, then goes out and populates the blood and the lymphoid organs throughout the body. Those lymphoid organs are secondary lymphoid organs. But the bone marrow is a primary lymphoid organ with regards to B cells, and the thymus is a primary lymphoid organ with regards to T cells. Now, clonal deletion is the way in which T cells and B cells are deleted. In the thymus, it's called thymic selection. But don't confuse that with clonal selection in terms of, when a clone is selected to proliferate because it's contacted its target antigen. So T cells, and this is quite different from B cells, even though the receptors are similar in molecular structure, the genes are quite similar. The T cell, they mature, undergo thymic selection, they migrate to the lymphoid tissues and populate them. They differentiate into fully mature effector T cells when they get activated by their appropriate antigen. Now, T cells that interact with their antigen will become either a T helper cell or a T killer cell. T killer cells are also called sometimes T cytotoxic cells. So there are two types of T cells. T helper cells, sometimes written like this with a H subscript, and T killer cells, or T cytotoxic cells, sometimes written like this, and quite often, actually, more often I would say, written like this. Okay. So you can get expansion of a T cell that has, by exposure to a particular single antigen, target antigen, you can get expansion of T helper cell populations and T killer cell population. The T helper cell has on its surface a molecule called CD4. It expresses on its surface a protein molecule called CD4. There are subcategories of T helper cells. It's always a little more complicated than you think. There are actually many, but these are the two major ones. There are T helper 1 cells, and there are T helper 2 cells. The T helper 1 cells produce cytokines like IL-2, interleukin-2, and interferon gamma. IL-2 is a growth factor for stimulated lymphoid cells. If a lymphoid cell is stimulated, activated by its antigen, it will start expressing on its surface IL-2 receptors. So it can respond to this cytokine which is a growth factor. It can bind IL-2 produced by T helper 1 cells and start expanding. T helper 2 cells are geared towards activating cells of the humoral immune response, B cells in other words. They help B cells to produce IgM and IgE. They'll produce a cytokine called IL-4. I'm not going to get into IL-4. The cytotoxic T cells, or TC cells, they have on their surface a molecule called CD8. These can be used as markers. CD4 positive cells versus CD8 positive cells. These cells are able to destroy any cell that is expressing a foreign antigen on its surface. Now that's important, particularly in viral infections, because if you have a eukaryotic cell in your body, there's its nucleus. If it gets infected with a virus, the virus will take over the cell, take over the cell's machinery. But one of the things the virus does also is it causes the expression of viral proteins on the surface of the cell. These viral proteins on the cell surface can be recognized by T killer cells that have the right T cell receptor that can respond to this. And they are able, these T cytotoxic cells, when they bind to something they're specific for will actually kill the target cell. In this way, they will kill infected cells, and they help in a tremendous way to prevent increase in infection and to kill off virally producing cells, host cells that are virally producing that were infected by the virus. So if you have, for example, a viral infection in your lungs and you have epithelial cells that have been infected with the virus, they will be taken over by the virus and start to produce more virus. They will express the viral proteins on the surface, and cytotoxic T cells can respond to that and kill those infected cells. They destroy the infected cells, and at the same time, often you'll have antibody molecules that can directly tag on to attach or bind to the virus and help other cells kill the virus or complement. So that's one very important part. The T helper cells, the most important thing here, I would say, is that they produce this growth factor that allows activated T cells and B cells to multiply, and the cytotoxic T cells that will bind to proteins on virally infected cells and kill them. And a lot of what I just talked about is shown in this diagram. You can go through it on your own. The one thing that I haven't talked about is the fact that macrophages are, and you have all kinds of different macrophages in your different parts of your body. So macrophage lineage cells are in fact quite important and very active as what are called antigen presenting cells, or APCs. What they do is they phagocytose and destroy and chop up. They'll phagocytose foreign structures, molecules, or like bacteria, for example, and they'll chop it up and they'll express it on their surface. Here you see expressed on the surface this foreign antigen fragment. It's always, and it'll be all over the surface, it's always expressed together with self molecules called MHC molecules. And helper T cell here, in this case, a helper T cell with the right receptor and the appropriate receptor recognizes that. So a lot of antigens need to be processed by antigen presenting cells and presented on the surface of the antigen presenting cells in order to get a proper immune response. Here you see interleukin-2, which is stimulating this cell to produce many helper T cells. Okay. There's interleukin-2 production. Okay, those are helper T cells and antigen presentation. Cell-mediated cytotoxicity I've spoken about. Here you have a infected cell in a host. It is infected with the virus. It is expressing viral antigen proteins or structures on its surface. It also, of course, has this molecule, which I told you is a tissue molecule. It's self-tissue. It always has to be together, self molecule and the foreign target antigen molecule always have to be together. The T cell receptor can, at least if you have a cytotoxic T cell with the appropriate receptor, it will bind and recognize, and it will release these molecules that will kill the infected cell. And if you kill off infected cells, then you're essentially stopping the increase in virus numbers. You're stopping these cells that are now viral factories, killing them off. So this is cell-mediated cytotoxicity, mediated by cytotoxic T cell. As I told you, there are many different types of cytokines. Of quite a few of them are called they've been given the name interleukins. Cytokines are soluble proteins that are released by cells mostly in the immune system and act as a signal for other cells. An important one is IL-1. IL-1 is produced by macrophages, not only macrophages that are triggered by LPS, although that's an extremely good and strong trigger, but also by macrophages that have phagocytosed to foreign particles. They'll produce IL-1 also. And IL-1 can help stimulate T helper cells. It also, of course, as you know, will increase the set point for temperature by the hypothalamus, but it stimulates T helper cell proliferation. T helper cells produce interleukin-2. It is a growth factor for all these different kinds of cells. Okay, it helps them proliferate themselves. So IL-2 produced by T helper cells will trigger more proliferation of T helper cells and T killer cells and B cells. I'm not going to get into IL-12. I already spoke about interferon gamma, a different kind of cytokine. It helps increase activity of macrophages so that they're even more active. They can be highly phagocytic. They can produce a lot of toxic compounds that could kill a microorganism. I've explained to you chemotaxis, movement of inflammatory cells towards the site of infection. You know, I talked about chemotactic factors like C3a and particularly C5a, which is an even stronger chemotactic factor. So these there are others that chemotactic factors that they're called chemokines. So, just wondering if we should know. I'm going to finish it off with part 2. So, you have immune tissue everywhere in your body, and its job is to try and prevent infection. A lot of the immune tissue is part of the innate immune system. Macrophages, granulocytes, the 3 different kinds of granulocytes, they form an important major part of the innate non-specific immunity. The rest is the adaptive immune response, lymphocytes, B cells, and T cells. If you look in the gastrointestinal tract, for example, you have, particularly in the last or third section of the small intestine, in the wall, you have mucosal associated lymphoid tissue called Peyer's patches. These are present in the submucosa of the GI tract. They contain a lot of dendritic cells, which are macrophage lineage cells that are very, very highly active as antigen presenting cells. These are professional antigen presenting cells. They're very good at taking up foreign molecules, chopping them up, and presenting them on the surface to other cells of the immune system. So there are dendritic cells. There are also T cells found in the Peyer's patches. I already introduced to you the idea that macrophages can present antigen, and there are all kinds of macrophages in the body. For example, in the liver you have Kupffer cells, in the lungs you have alveolar macrophages, in the brain you have microglia. You have splenic macrophages, you have peritoneal macrophages, and you also have dendritic cells, which are found in the in association with epithelial tissue. And dendritic cells, as I already said, are even more active than other types of macrophage lineage cells at taking up foreign molecules, chopping them up, and presenting them on the surface. So they're very, very good at presenting antigen. They are often called professional APCs, or antigen presenting cells. Now, what I didn't talk about are that the is that what I haven't talked about, and these are huge topics, but I'll try and give you the very fundamental basics. There are cells in your body that are capable of being, they are already active. They're part of the innate immune system. They include macrophages and natural killer cells. Now, these cells can be activated further to be extremely active. So they're stimulated by phagocytosis or by cytokines like interferon gamma. These cells also are stimulated. They are active on their own, but can be further stimulated by interferon gamma. And these are able to kill target cells. So their macrophages can increase their phagocytic activity. They can increase their production of toxic oxygen radical molecules, and they can actually kill tumor cells, as can natural killer cells. Natural killer cells will destroy virus-infected cells and also tumor cells. And it's not here, but macrophages also can kill tumor cells. They are also quite good at killing tumor cells. These are non-specific cytotoxic cells, unlike the cytotoxic T cell that has a particular receptor on its surface and will kill cells that have foreign antigen on their surface, like a virally infected cell. These cells will kill non-specifically tumor cells and virally infected cells, etc. I'm not sure that I want to talk about this. I mean, it's a fairly simple concept. Some antigen structures, some foreign antigen structures can activate B cells without the help of a T cell. These type of antigens are called T-independent antigens. They don't need help, they don't need help from T cells. The B cells do not need help from the T cells. The B cell will be able to respond to a T-independent antigen. The reason is the antigens, these type of antigens, T-independent antigens, have many repeating subunits of the same epitope. And that includes polysaccharides and lipopolysaccharides. Remember, polysaccharides are long carbohydrates. Lipopolysaccharides are part lipid, part polysaccharide, and bacterial capsule. These are all able to act as T-independent antigens and activate B cells that have the right responding molecule on their surface. And then there are T-dependent antigens, and T-dependent antigens must be presented by an antigen-presenting cell. The antigen is taken up by the antigen-presenting cell and then presented on the surface together with MHC, which is a self molecule. It's a tissue molecule. So foreign antigen plus self will activate a T helper cell that has the appropriate T helper cell receptor on its surface, so they can react to that, and T helper cell produces IL-2, and the IL-2 will activate a B cell that also has the right kind of antibody or receptor, antigen receptor, which is an antibody on its surface. I wouldn't worry too much about, just know that I wouldn't worry about the steps here. I would just be aware that there are T-independent antigens and there are T-dependent antigens, and the T-independent ones act because they have this repeated structural motif or repeated structural characteristic, the same antigen lined up one after another, whereas the T-dependent ones, in terms of B cell response, we're looking at triggering a B cell response. T-dependent antigens have to be phagocytosed, then presented by antigen-presenting cells, and then a helper cell can respond to that presented antigen and help activate a B cell. Okay, so we have 2 slides left. This is an important slide in that it talks about acquired immunity. We acquire immunity in a number of different ways, and acquired immunity is occurring all through each individual's life in response to the foreign molecules or foreign target antigens that they're exposed to. But they can occur in different ways. They can occur naturally, so they can be naturally acquired, and it could be an active process or it could be a passive process. So naturally acquired active immunity is the result of an infection. The microorganism gets into you, you have an active infection. That's a natural process. It triggers an active immune response. It'll trigger, if it evades the innate immune system, then it'll trigger the adaptive immune system to respond. The T cells and or B cells that have the appropriate receptor will interact with that target antigen. They will be activated. They'll start to proliferate. The B cells will proliferate and start secreting antibody. The T cells will start, T helper cells will secrete IL-2. That'll help more T helper cells to proliferate of that type, and also help the T killer cells to proliferate, the ones that have the right receptor for the foreign antigen, and those can kill infected cells. So that is a natural process, and it is an active process because the cells with the appropriate receptor have to interact with the antigen and have to multiply. We also have naturally acquired passive immunity. In this case, naturally acquired passive immunity refers to the immunity that is passed on naturally to a fetus or via antibody crossing the placenta. Remember that some antibodies can cross the placenta and others cannot. IgG can cross the placenta. That's a passive process. The fetus is not making the antibody, it's just receiving it passively from the mother's serum or plasma. The other way that immunity can be naturally and passively acquired is via colostrum. Colostrum is the very first milk that's produced following birth of the baby. The mother's milk is very rich in IgA class antibodies, and this helps to passively give the newborn immunity by breastfeeding and taking up those antibodies. So we have naturally acquired active as a result of infection, or naturally acquired passive as a result of transfer of the antibody molecules. Here, the naturally acquired active, there are actually B cells that are activated and proliferate, and also T cells. And here it's all about just antibody passively being transferred over. Now the other way that we can acquire immunity, and this is developed much more recently over the past couple of hundred years, I guess, artificially acquired active immunity. In this case, rather than infection, which is a natural process, the artificial part is the vaccination. You are vaccinated or immunized, same idea, by injection of antigen. So an antigen associated with a particular microorganism, pathogenic microorganism, is injected into you. It doesn't give you the disease, but it exposes your immune system to molecules that are part of that microorganism, and it triggers in you an active immune response. You become actively immune because you have T cells activated, or B cells, or both. And the fourth way is that you can acquire immunity is artificially acquired passive immunity, where the person is artificially injected with a particular antibody. So here again, it's not the cells that are being activated that will provide you with memory and the ability to respond, but just the antibody. So it's not permanent. Passive is not permanent. Just involves antibody, and the antibody is injected, and we talked about that. It can be done for snake bites. It's often used if there's just not enough time for your immune system to respond. You know, things like infection if you get exposed to rabies virus infection progresses so quickly that if your exposure did infect you, then you'll be dead before you have a naturally acquired active immune response. And so what they do is they give you artificially acquired passive immunity by injecting you with antibodies against rabies virus. They also might vaccinate you to boost your own system, but after all, the antibody won't hang around forever. But they also do that for snake venom. The snake venoms are highly toxic, and even people who survive them without treatment are often have long-term effects. So they'll give antibody specific active. They'll give the artificial part is the injection. Artificially acquired passive immunity, so you have the antibody, and they're always giving antibodies that will inactivate the toxin in the venom. And so we get to the last slide, and the last slide is a summary of what I was just talking about: acquired immunity, different kinds of acquired immunity. There's naturally acquired, in which you get infected, or you naturally acquired active, in which you get infected and the antigens are entering your body naturally along with the microorganism, so that your own immune system produces antibodies and T killer cells or T cytotoxic cells. Passive involves passively acquired, naturally acquired but passive acquisition of the antibodies from the mother across the placenta to the fetus, or via the mother's milk into the newborn. Artificial systems involve injection, either triggering an active immune response or allowing for passive immunity. So artificially acquired active immunity is when the antigens are introduced into the body by vaccination. You produce antibody and T cell responses, or passively you're putting in known antibody, immune serum antibody into sometimes it's monoclonal, not a serum antibody. Okay, those are the four kinds: naturally acquired active, naturally acquired passive, artificially acquired active, and artificially acquired passive immunity. And that's the end of lecture 17, part 2, and the end of explanation on the adaptive immune response.